Good day to everyone. My name is LEE ZHI XUAN. So this video I will explaining about the mechanism of differential centrifugation. So this is the last topic belong to the prokaryotic cellular component. So hopefully you guys can hear it and about what is the differential centrifugation. So normally this is the experimental technique to want to analyze the component of the organelles. If you want to analyze any component of the organelle, you want to make a research or you need to do an experiment, you need to using this differential centrifugation. It is a technique to separate the particle using the centrifugation force. In general, it is that using the machine to rotate the mixture very fast. It's very fast, like using the bladder or using the centrifugal. So it like that. They are pushing, rotate, very fast one. You need to using the machine one. Our human hands cannot rotate like that. It is very fast. Normally in 1 second, in 1 minute, they can rotate to 20,000, 30,000 times one. 1,000 times one. 1,000 times one. So they rotate very fast. Very fast. Okay. So this is the why we want to do this technique. Normally, this one is to separate the cellular component. You still remember when you learn about organelle, ribosome, mitochondria, cytoplasm, chloroplast, all that are organelles. So when you are the scientist, you want to do the research only the certain organelles. Only the certain organelle. Maybe you only want to do a research about the mitochondria. So you need to isolate mitochondria. So how do you isolate mitochondria only? I only want mitochondria. Others organelles I don't want. Like ribosome I don't want. Golgi apparatus I don't want. So this is the basic differential centrifugation technique lah. So this one is just a basic technique. So if you want to refer to very deep, every type of the technique have every different type of the isolation of particle or organelles have different technique. But this is just a basic, a normal procedure that you can refer that you need to know. Okay, so the most important thing is that you first step is you need to have a tissue culture. Any cell, like our human, you need to have a skin cell, or maybe you need to have the blood, or maybe you need to have a muscle cell. Any cell, you need to have a sample first, cell sample. This one. After the first step is you need to homogenize it. Homogenize, meaning that you need to break down the cell first. Because I don't want the cell itself, I only want the component inside the cell. So you need to kill the cell first. You need to kill the cell. So you need to break down the cell. So normally, not necessarily plant cell, any cell. So normally the cell is you need to cut it, and place it in the cold and isotonic buffer solution first. The main key point is to cold, low temperature, and also isotonic, distilled water. You cannot immerse in salt solution. You cannot immerse in very highly sweet solution like that. Yeah, cannot. You need to not distilled water, isotonic solution. So why low temperature can deactivate your hydrolytic enzyme to minimize the self-digestion. So you need to deactivate your hydrolytic enzyme. And also the second one is isotonic solution can maintain the shape and size of the organelles. Although you need to break down or you need to kill the cell, but you don't want to destroy the organelle. If you place in the hypotonic solution or hypertonic solution, you not only kill the cell, but you also able to kill the organelles. You don't want. You need to maintain the organelle first. And final one, buffer solution is to minimize change in pH to prevent the nutrition of protein enzyme release. So one of the components is maybe when you want to do the experiment about the protein enzyme inside the cell. This one is also you need to alert it. If you put in the very low pH or high pH value, you not only kill the cell, but you also able to kill the enzyme within the cell. We don't want. We need to maintain it. Okay, so once you already do this experiment, then you need to homogenize it. You need to break the cell open to release all the components inside the cell. Also, all the organelles and also protein, enzyme 什么 or you need to break the cell so that release it, we will have a homogenate like that. You already break up the cell, you already have this homogenate. Okay, once you already have a homogenate, you need to filter it by using the muslin cloth, using any type of cloth lah. Sometimes they using filter funnel, sometimes you using the cloth, any acceptable apparatus you can use, but you need to filter lah. And separate the big fragment away from the small fragment. So this one is based on the experiment purpose that you need one. Either you want to a big fragment or small fragment lah. So for now, we need to take the very small fragment. Okay. So the particles are suspended in the liquid media and placed in the centrifugal like that. This one is we call a centrifugal lah. Okay. Then it will place to the rotor or spun or centrifugal machine, and they will spun at very high speed, at the different speed. And the filtrate is then centrifuged at different speed and separate to the component of the cell. Okay, so once they already go to the centrifuge, they will run at different speed. So normally first speed, so you can see here, first speed they run at 1,000 grams, meaning that this is the 1,000 times the force of gravity for 10 minutes. Okay, so you just imagine that it is very fast, very fast, already very fast, 1,000 G, very fast. Okay, now then you will see that they will have a two layer in your mixture. One is we call as supernatant, which is a liquid mixture like that at the upper part, and another one is we call as pellet, solid pellet. So when you do this, what does it mean? Meaning that this is the first time. Meaning that at the lower part, normally the larger organelles, especially nucleus, and also any cell that breathes, the cell myard of the cell, or any larger protein molecule, they were inside the pellet. And other organelles were still in the still in your supernatant, meaning that only the nucleus is inside your pellet. Nucleic acid DNA, RNA, all that is in your pellet. But other organelles still inside your supernatant. So if you are doing the experiment, want to do a research about the nucleic acid, then you already can take this. You will remove all the supernatant and then you directly use that pellet for your research purpose. But maybe you don't want pellet. I want maybe Golgi apparatus, or maybe I want mitochondria. Okay, if you want to mitochondria, what you need to do? You need to transfer this supernatant to another centrifuge. When you transfer it, you will go to another new tube and then you centrifuge again. And now you will centrifuge more high speed and more time, longer time and very high speed. Once you already do it, you also will get these two. You also will get supernatant and also pellet. Now your pellet, what are the content of your pellet? Your pellet will containing either mitochondria or chloroplast. So normally in plants they will containing two mitochondria and chloroplast, but in animals of course it containing mitochondria only. So your organelle inside your pellet containing mitochondria and also chloroplast. And others one Golgi apparatus, all that still remain in your supernatant. So if your research is about mitochondria or chloroplast, then you can already use this pellet and throw away the supernatant and use the pellet. But you don't want, you still want about the Golgi apparatus. Okay, you still repeat the step. You we transfer this supernatant to another tube again and centrifuge again, rotate again, and very high speed and also more longer time. You see, already 1 hour. Now your contain will containing other kind of organelles Golgi apparatus, endoplasmic reticulum, all that. And if you want ribosome is very small, the smallest organelles. Then you repeat the step again. And then centrifuge more high speed and also more longer time. Finally, you will get ribosome, just like that. So this is a overall procedure about the differential centrifugation. This is the explanation that you need to write in the question, in the answer. Okay, so that's all for this video. So hopefully you guys can understand it. What is the basic technique about the differential centrifugation? So thank you for your watching and bye.